All right, welcome to Chapter 6, Genetic and Developmental Diseases. This is one of those chapters that sounds intimidating right out of the gate, but I promise. We're going to break it down into pieces that actually make sense. This chapter is really about how things can go off script during development, whether that happens at the level of a single gene, an entire chromosome, or because the developing embryo or fetus is exposed to something it shouldn't be. We'll talk about genetics, yes, but not in a Punnett Square freshman bio kind of way. We're looking at genetics through a clinical lens. What went wrong? When did it go wrong? And how does that show up in real patients in real specimens? As histotechnology students, this matters more than you might think. You're not just cutting slides, you're often looking at the downstream effects of these disorders in tissue. Fibrosis, abnormal architecture, missing structures, or things that just don't look the way they're supposed to. Also, and this is important, many of the conditions we'll talk about are congenital, genetic, or developmental, but those words are not interchangeable. By the end of this chapter, you should be able to use them correctly and gently correct someone else when they don't. I always tell students if this chapter feels like a lot, that's because human development is a lot. It's actually kind of amazing that it works as often as it does. As we go through this chapter, I'll keep pointing out places where this shows up in the lab. Placenta specimens, pediatric biopsies, autopsy findings, and even things you might see incidentally years later in adult tissue. A fun fact to start us off, human development takes about 18 years to fully complete, so technically adolescence is just the longest quality control phase ever. Before we go anywhere else, we have to start with DNA, because everything in this chapter eventually comes back to it. DNA is a double helix made up of 4 nitrogenous bases: adenine, thymine, guanine, and cytosine. You already know the base pairing rules: A with T, G with C, so we're not going to relive freshman biology trauma here. The key thing to remember is that DNA encodes all of our genes. Genes are just specific sequences within that DNA that carry instructions, usually for making proteins. And those genes don't just float around independently. Multiple genes live together on chromosomes, which is why when something goes wrong at the chromosome level, it often affects more than one trait. And this visual is doing a lot of work for us. It shows how DNA is packaged from a loose strand into tightly wound chromosomes inside the nucleus. Nature really said, space-saving mode. When you're looking at genetic diseases in tissue, you're rarely seeing a DNA problem directly. You're seeing the consequences of incorrect instructions playing out at the cellular and tissue level. If you stretched out all the DNA in one human cell, it would be about 6 feet long, which feels unnecessary, but here we are. This slide introduces one of the most important distinctions in genetics: genotype versus phenotype. Your genotype is your genetic blueprint, your collection of genes. Think of it as the instructions. Your phenotype is what actually shows up. These are the observable traits. So morphology, development, biochemical function, physiology, basically what the instructions produce after they interact with the real world. And that last part matters. Genotype does not automatically equal phenotype. Environment, timing, epigenetics, and a whole lot of biological chaos all influence how genes are expressed. So 2 people can have the same mutation and look very different clinically, and you'll see that theme come up a lot in this chapter. This is why genetics is fascinating and frustrating in equal measure. Now we zoom out from individual genes to the chromosome level. Karyotyping is the study of chromosomes, specifically their number, size, shape, and structure. Each human somatic cell normally contains 46 chromosomes, arranged as 23 pairs. 1 chromosome in each pair comes from each parent. Of those pairs, 22 are autosomes, and 1 pair is the sex chromosomes. So when you see notations like 46, XY or 46, XX, that's shorthand for a numerically normal karyotype. This slide is laying the groundwork for when we start talking about trisomies, monosomies, and chromosomal syndromes. This is the what normal looks like reference point. Karyotypes aren't something you'll perform as histotechs, but understanding them helps you interpret pathology reports and clinical correlations, especially in prenatal and pediatric cases. This is one of those slides that looks harmless but carries a lot of weight. Haploid means having 23 chromosomes. This applies to gametes, meaning sperm and ova. Diploid means 46 chromosomes. That's your somatic cells. Euploid means any exact multiple of the haploid number. So diploid cells are euploid. Triploid would also technically be euploid, even though clinically that's a problem. And aneuploidy is when the chromosome number is not an exact multiple of 23. This is where we start running into clinical syndromes. Then we narrow it down further. Monosomy equals one chromosome is missing, and trisomy equals one extra chromosome. These terms will come up constantly, so being fluent in them will save you brain energy later. If genetics had a vocabulary quiz, this slide would be half of it. Next, we zoom back to the gene level. A gene is a specific sequence of DNA that codes for a trait, often through protein production, and it's important to remember that more than one gene can influence a single trait. Polymorphism refers to the presence of multiple alleles at the same genetic location with noticeable differences in phenotype. This is normal human variation, not necessarily disease. And an allele is simply a version of a gene at a specific locus on a homogeneous chromosome. So when we talk about inherited conditions, we're really talking about which alleles are present and how they interact. This slide is mercifully the last of the vocabulary marathon, but it's important. Homozygous means two identical alleles for a gene. Heterozygous means two different alleles for that gene. And hemizygous means only one copy of a gene is present. This last one is especially important for sex-linked disorders. Males are homozygous for most genes on the X chromosome because they only have one X and one Y. So if there's a recessive mutation on the X chromosome, males don't have a backup copy. And that's why conditions like hemophilia and Duchenne muscular dystrophy show up the way they do. This term will quietly explain a lot of inheritance patterns once we get there. Before we get into specific diseases, we need a shared language for how genes behave. Alleles are basically different versions of the same gene, and how they express determines whether or how a disease shows up. Autosomal dominant means one copy is enough. If the mutation is present, the trait or disease is expressed, no backup copy. Autosomal recessive requires two copies, one from each parent. These are the ones where people can be carriers and not even know it. And then we have sex-linked traits, where the gene is on the X or the Y chromosome. These show up differently depending on chromosomal makeup, not gender identity chromosomes. Important distinction. Dominant doesn't mean worse, and recessive doesn't mean mild. Genetics is not a personality quiz. This is where genetics stops being binary. Codominance means both alleles are fully expressed at the same time. A classic example is AB blood type. A and B are both doing their thing unapologetically. Incomplete dominance is different. Instead of both showing up clearly, you get a blended phenotype. Not red, not white, but pink. The flower images here are doing the heavy lifting. Think side by side for codominance, blended smoothie for incomplete dominance. Your hair color is actually a great real-life example of incomplete dominance, which explains why your childhood photos don't match your driver's license. All right, this slide introduces two terms that students love to mix up: penetrance and expressivity. So we're going to slow this down just enough to make it click. First, penetrance. Penetrance is about whether a gene shows up at all. It's the proportion of people who carry a specific genotype and actually express the associated trait. So if a disease has 100% penetrance, everyone with the genotype shows the phenotype. No mystery, no surprises. If penetrance is incomplete, some people carry the gene and you'd never know it clinically. Penetrance asks, did the gene speak up or stay quiet? Now, non-penetrance is when the gene is present, but there's no abnormal trait expressed. This can happen even with autosomal dominant genes or with individuals who have two autosomal recessive alleles but don't show disease. So yes, genetically affected, clinically fine. Genetics loves exceptions. Worthless but comforting knowledge. This is why family histories sometimes look confusing. The gene may be there, but it's not always loud. Next up is expressivity, which answers a different question. Expressivity is about how strongly or how severely a trait is expressed in individuals. Who do show the phenotype? Everyone has the gene, everyone shows something, but the degree varies. One person might have mild symptoms, another severe, another somewhere in between. Same genotype, different presentation. Think of it this way. Penetrance is the on-off switch. Expressivity is the volume knob. And this distinction matters, especially in clinical genetics and pathology, because it helps explain why two patients with the same mutation can look very different in real life. So when you're thinking about genetic disease patterns, remember, penetrance equals does it show up at all, and expressivity equals how intense is it when it does. Yeah. All right. This slide clears up one of the most common vocabulary mix-ups in pathology and genetics: hereditary, congenital, and familial. These terms get used interchangeably in casual conversation, and they absolutely should not be. First up, hereditary. Hereditary simply means derived from one's parents, passed down through genes. You also hear this referred to as familial, but keep that word in your back pocket for a second. Key idea here is hereditary equals genetic inheritance. If it came from mom, dad, or both, genetically, it's hereditary. Now let's talk about congenital, which is where people often go sideways. Congenital does not mean genetic. Congenital only means present at birth. That's it. No DNA requirement. Some congenital diseases are genetic, sure, but some are absolutely not. Classic examples: congenital syphilis, present at birth? Yes. Genetic? No. That's an in utero infection. And here's the flip side. Not all genetic diseases are congenital. Take Huntington disease. Completely genetic, completely hereditary, but symptoms usually don't show up until someone's 20s or 30s. So the gene is there at birth, it just waits patiently, like a very rude house guest. Now let's bring in familial diseases, which sounds straightforward but has nuance. Familial diseases are conditions where multiple family members are affected by the same genetically or chromosomally based disease. But, and this is important, genetic does not automatically mean familial. Many people with genetic diseases have no known family history. Why? Two big reasons. The disease may be recessive, like cystic fibrosis. Carriers don't show symptoms. Or the disease may result from a new germ cell mutation, meaning it starts with that individual. You can absolutely be the first one in your family to win the genetic lottery. Sorry. So here's the takeaway. Hereditary equals passed down genetically, congenital equals present at birth, and familial equals seen in multiple family members. And none of these terms are synonyms, even though people use them like they are. All right, let's start with the cell division we all know, love, and sometimes take for granted: mitosis. Mitosis is your classic copy-paste situation. One cell divides to produce two genetically identical daughter cells. Same DNA, same chromosome number, same instructions, no surprises. This happens in somatic cells, meaning basically everything that isn't making babies. So skin, liver, intestinal epithelium, if it needs routine replacement, repair, or growth, mitosis is doing the heavy lifting. From a histotech perspective, this is why tissues maintain their architecture. Cells die, cells divide, and the tissue keeps its identity. Mitosis is the reason your H&E slides don't turn into abstract art overnight. A fun but mildly useless fact, your intestinal epithelium replaces itself roughly every 3 to 5 days, which means the cells lining your gut right now were not invited to your last bad life decision. Also important clinically, when mitosis goes off the rails, that's where neoplasia starts knocking. Uncontrolled division, loss of regulation, and suddenly pathology has job security. Now let's talk about meiosis, which is dramatically different. Meiosis is the process that produces germ cells, sperm and ova, and its entire purpose is to cut the chromosome number in half. Each resulting cell ends up with 23 chromosomes, not 46. Why? Because fertilization is a reunion tour. Egg plus sperm restores the normal diploid chromosome number and creates the zygote. Meiosis happens in 2 rounds, meiosis 1 and meiosis 2, but DNA only replicates once. The real magic and chaos happens in meiosis 1, when homologous chromosomes pair up, exchange material, and then separate. This is where genetic variation comes from. Independent assortment and crossing over. Translation: meiosis is why siblings look related but not identical, and why family photos are emotionally complicated. A worthless but entertaining fact, the genetic recombination that happens during meiosis means that the odds of 2 siblings having the exact same genome, excluding identical twins, is astronomically low. Nature is a maximalist. This slide basically says what your brain is already screaming. Mitosis, 1 division, 2 identical daughter cells, full chromosome set, growth, repair, maintenance. Very boring, yet very dependable. Meiosis, 2 divisions, 4 genetically unique cells, half the chromosome number, reproduction and genetic diversity, chaotic neutral energy. I absolutely love this cartoon because it's accurate. Mitosis is Attack of the Clones, and meiosis is very much Dare to be Different. From a pathology standpoint, mistakes in meiosis are how we get aneuploidy, trisomies, monosomies, and chromosomal disorders that show up later in this lecture. Mitosis mistakes usually lead to cancer. Meiosis mistakes usually lead to genetics consults and various serious conversations. Different processes, different consequences, same chromosomes. So as histotechnologists, why do we care? Because understanding how cells divide explains why tissue looks the way they do, both normal and abnormal. When you see increased mitotic figures, abnormal spindles, or disorganized tissue architecture, you're not just seeing cells dividing, you're seeing regulation fail. And when you're dealing with chromosomal abnormalities, prenatal specimens, or pediatric cases, meiosis is usually where the story began, long before the specimen ever reached your lab. All right, now we're moving from gene level problems to chromosome level chaos, and this is where things get structurally interesting. When we talk about chromosomal abnormalities, we're not talking about a typo in a gene. We're talking about entire chunks of chromosomes being rearranged, copied, lost, or swapped like genetic Lego pieces. So first up is duplication. This is exactly what it sounds like. A fragment of a chromosome gets copied, so now there's extra genetic material hanging around. And more DNA does not automatically mean better. Cells like balance, not bonuses. Next is deletion, which is usually more problematic. This is the loss of a segment of a chromosome during meiosis. If that deleted region contains essential genes, and it usually does, the downstream effects can be significant. Then we have translocation, where pieces of chromosomes decide they want to live on a different chromosome entirely. These exchanges happen between non-homologous chromosomes, which is important. Sometimes translocations are balanced and the person is mostly fine until reproduction enters the chat. And this is why karyotypes in cytogenetics matter. Structure tells a story that sequencing alone might miss. Some worthless but fun knowledge. Some chromosomal translocations were first identified in cancer cells before we ever understood their genetic consequences. Cancer, once again, teaching us biology whether we asked or not. This slide introduces one of the most important concepts in cytogenetics, non-disjunction, which is a fancy way of saying chromosomes fail a very basic separation task. During normal meiosis, homogeneous chromosomes are supposed to separate cleanly. When non-disjunction occurs, they don't. Instead, both chromosomes go into one gamete, and the other gamete gets none. Now here's where it becomes clinically relevant. If fertilization happens with one of those gametes, the resulting zygote ends up with too many chromosomes, which is trisomy, or too few, which is monosomy. And this is not subtle. Cells are very sensitive to chromosome number. Gene dosage matters. And yes, non-disjunction is strongly associated with maternal age, but it's not exclusive to it. Biology loves probabilities, not guarantees. This is why prenatal screening, FISH, and karyotyping exist. You're not just counting chromosomes, you're predicting viability. Let's talk outcomes, because not all chromosomal abnormalities are compatible with life. Monosomy involving an autosome is almost always lethal. Losing an entire chromosome means losing way too much genetic information. The cell cannot compensate. Trisomies, on the other hand, can sometimes permit survival, but usually with severe consequences. With the notable exception of trisomy 21, most autosomal trisomies result in infants who survive only a short time after birth. And this is where language matters. These outcomes aren't about extra or missing chromosomes being good or bad. They're about gene dosage imbalance. Cells are calibrated systems. Throw off the ratios and development struggles. This is also why you'll hear the phrase incompatible with life in pathology reports. It's not dramatic, it's descriptive. Some worthless knowledge. Humans tolerate sex chromosome abnormalities far better than autosomal ones. Turns out we're much more forgiving when it comes to X's and Y's. Mosaicism is one of my favorite topics because it reminds us that biology does not follow clean rules. Mosaicism occurs when mitotic errors happen after fertilization, leading to two or more populations of cells with different chromosomal complements in the same individual. So instead of every cell carrying the same abnormality, you get a mix, some normal, some not. This usually happens early in development, during cleavage of the fertilized ovum, but it can also occur later in somatic cells. And here's the key point. The severity of symptoms depends on where those abnormal cells end up and how many there are. Tissue distribution matters. Mosaicism commonly affects sex chromosomes, which is why you'll see variable presentations in conditions like Turner syndrome. From a diagnostic standpoint, mosaicism can be tricky. One tissue sample may look normal while another tells a very different story. As a histotech, sampling matters. Tissue choice matters. And this is why normal result doesn't always mean no abnormality. All right. Let's talk about mutations. And no, not the superhero kind. Although honestly, some of them do have superpowers, just not always the good kind. A mutation is a permanent change in the DNA sequence. Permanent being the key word here. This isn't a typo that autocorrect can fix later. Now, where that mutation happens matters a lot. If a mutation occurs in somatic cells, meaning your everyday body cells, it is not inherited. These mutations stay with you, not your future offspring. That's why somatic mutations are heavily involved in cancer development and sometimes congenital malformations, but they don't get passed down the family tree. On the flip side, mutations in germ cells, the sperm or the ova, are transmitted to the next generation. These are the ones that can result in inherited genetic diseases. Think of this as the difference between damage to a photocopy versus damage to the original file. Worthless but fun fact, every time a cell divides, it has to copy about 3 billion base pairs. The fact that it usually gets it right is honestly impressive. From a histotech perspective, remember, mutations often show up as abnormal protein expression, altered cellular architecture, or unexpected staining patterns, which means we are sometimes the first ones to notice something is off. This is one of my favorite topics as well because it proves that DNA is dramatic, but flexible. Epigenetic alterations are heritable chemical modifications to DNA or chromatin that do not change the DNA sequence itself. So the letters stay the same, we're just changing how loudly they're read. Think of epigenetics as a dimmer switch, not an on and off button. The gene is still there, but it might be whispered or shouted. These changes often involve things like DNA methylation or histone modification, which affect how tightly DNA is packed. Tightly packed DNA, you ask? Harder to transcribe. Loosely packed DNA, gene expression party. What's especially important here is that epigenetic changes can be influenced by environmental factors, things like nutrition, stress, toxins, even aging. So yes, your lifestyle choices can absolutely leave molecular receipts. And here's a fun fact you didn't ask for. Identical twins start life with nearly identical epigenomes, but over time they become more epigenetically different, especially if they live different lifestyles. From a lab standpoint, epigenetic changes help explain why two patients with the same mutation can have very different disease severity. Same gene, different volume level. This slide is your gentle reminder that biology is messy, and sometimes we just don't have all the answers. About 2% of newborns are born with congenital anomalies. That number is higher than most people expect, and it's why developmental pathology is such a big deal clinically. Now let's break down the causes. Embrace yourself because the biggest category is deeply unsatisfying. 65% are unknown. Yes, unknown. Science shrug. About 20% are genetic, meaning single gene or multifactorial inheritance patterns. Roughly 5% are chromosomal, which includes things like aneuploidies and structural abnormalities. And about 10% are environmental, exposures like infections, drugs, toxins, or nutritional deficiencies during critical windows of development. A worthless but memorable fact, the embryonic period is so sensitive that the same exposure can cause completely different outcomes depending on the exact week it occurs. For histotechnologists, this variability is important. Developmental diseases don't always follow neat textbook rules, which is why correlating histologic findings with clinical history is absolutely essential. And finally, this slide is a good reminder to approach these topics with scientific precision and human compassion. Behind every percentage point is a real patient and a real family. All right, this slide is really about how genetic diseases are categorized, which helps us understand why they behave the way they do and why some are easier to predict than others. First up, monogenetic defects. These are the one bad gene is all it takes situations. A single abnormal gene is responsible for the disease, and these follow classic Mendelian inheritance patterns. Autosomal recessive, autosomal dominant, X-linked, the whole genetics throwback you probably remember from earlier coursework. Good examples here are phenylketonuria, or also known as PKU, and sickle cell anemia. One gene, one main problem, though the downstream effects can be pretty significant. From a lab perspective, these are often the disorders we can detect early and manage well if screening is done correctly. A quick aside, Mendel would be thrilled that his pea plants are still paying rent in modern medicine. Next are polygenic defects, which are a bit messier because biology loves chaos. Here, multiple genes are involved. and often there's an interaction with environmental factors. This means genetics loads the gun, but environment often pulls the trigger. Conditions like diabetes or esophageal cancer fall into this category. You might inherit susceptibility, but lifestyle, exposures, and timing all play a role. These are harder to predict, harder to prevent, and harder to pin on a single cause, which is why risk assessment is such a big deal here. This is why people can say it runs in my family and still not develop the disease. Genetics isn't destiny. And finally, we have chromosomal diseases. These aren't about a single gene malfunctioning. They're about structural changes in chromosomes themselves. That could mean extra chromosomes, missing ones, or pieces that are rearranged, deleted, or duplicated. This is where karyotyping and cytogenetics come into play. The issue isn't what the gene says, it's whether the gene is even there, in the right place, and in the correct number. So when you zoom out, these three categories really represent three different levels of genetic disruption: gene level, multi-gene plus environment, and chromosome level. All right, now we're officially entering Mendel's house. P's optional. When we say monogenetic or single gene diseases, we're talking about conditions caused by one faulty gene doing one very unhelpful thing. The key here is that these diseases follow predictable inheritance patterns, which is why Mendel still gets invited to biology parties centuries later. These diseases are classified based on where the gene lives, on an autosome or a sex chromosome, and how bossy it is, meaning dominant or recessive. That's where our four inheritance patterns come in: autosomal recessive, autosomal dominant, sex-linked recessive, and sex-linked dominant. You don't need to memorize them all right now. We're going to unpack each one slowly and pain-free. Think of this slide as the table of contents. The chaos comes later, but in an organized way. Worthless but fun knowledge: Gregor Mendel didn't even know what a gene was. He just knew peas were snitches. A few big picture trends to anchor us before we dive into specifics. First, autosomal diseases are more common than sex-linked ones. Why? Because autosomes make up the bulk of our chromosomes, so statistically, they're more likely to cause trouble. Second, recessive disorders are more common than dominant ones. That's because recessive mutations can quietly hang out in carriers without causing disease. Kind of like that one coworker who does nothing but never gets fired. Another important concept: many of these diseases show incomplete penetrance and variable expressivity. Translation: not everyone with the mutation shows symptoms, and not everyone who does show symptoms looks the same. Biology, again, loves chaos, but with rules. Autosomal dominant disorders only need one defective copy to show up. No backup plan required. Autosomal recessive disorders need two defective copies, which is why carriers are usually unaffected. And finally, single gene defects don't just cause random symptoms, they reflect what protein is broken. Structural protein: expect physical issues. Enzyme defect: metabolic chaos. Receptor problem: signaling disasters. This is why enzyme deficiencies show up so dramatically in metabolic diseases. If the protein doesn't work, substrates pile up like unprocessed tissue blocks on a Friday afternoon. Autosomal dominant disorders are the overachievers of genetics. One copy is enough to make itself known. Typically, the affected individual is heterozygous, and usually one parent is affected as well. That family history is often a big red flag during diagnosis. These disorders affect all sexes equally because autosomes don't care about gender. They're equal opportunity disruptors. One hallmark feature is later onset, often in adulthood. This is why conditions like Huntington disease don't show up at birth, even though the mutation is there from day one. Clinical features can vary widely because penetrance and expressivity are doing their thing. Plus, environmental factors like lifestyle, exposures, and sheer bad luck play a role. Many autosomal dominant disorders involve receptor proteins or structural proteins, which explains why their effects tend to be systemic rather than isolated. Bottom line, Autosomal dominant disorders are predictable in inheritance, unpredictable in presentation, and very good at keeping genetic counselors busy. A fun and slightly dark fact, some autosomal dominant disorders persist in populations because symptoms appear after reproductive age. Evolution never got the memo. Huntington's disease is the classic teaching example of an autosomal dominant disorder. It has complete penetrance, meaning if you inherit the mutation, the disease will manifest, no maybes. It results in progressive destruction of neurons, particularly in the basal ganglia, which explains the movement and cognitive symptoms. Each child of an affected parent has a 50% chance of inheriting the disease. On the right, you're seeing the effects of neuronal loss, especially enlargement of the lateral ventricles due to surrounding brain atrophy. This is also one of the most emotionally complex genetic diagnoses because people often know they carry the mutation long before symptoms begin. Manifestations of the disease do not develop until middle age. Till a middle-aged person has already likely had children and can transmit the disease. This is another autosomal dominant disorder, but with a completely different mechanism. The mutation affects the LDL receptor, meaning the liver can't adequately clear LDL cholesterol from circulation. The result is chronically elevated cholesterol, which leads to premature arteriosclerosis, often at a very young age. These yellow plaques you see are xanthomas and phantylasmas, lipid-filled macrophages in skin and tendons. These lipid-rich lesions are a great reminder that metabolic problems leave physical footprints. Familial hypercholesterolemia is a condition that helped drive early cholesterol research and is one reason statins exist today. All right, let's talk about Marfan syndrome, which is one of these conditions that pathologists absolutely love because the genetics, the structure, and the clinical picture all line up very neatly. Marfan syndrome is a connective tissue disorder, and it primarily affects tissues that depend on elastic fibers. That's why the biggest hits are to the skeleton, eyes, and cardiovascular system. Basically anything that needs to stretch and not immediately fall apart. Genetically, this is an autosomal dominant disorder, so one defective copy of the gene is enough to cause disease. The prevalence is about 1 in 5,000, which means statistically there are more people with Marfan syndrome than people who claim they don't need sleep. The root cause is a defect in fibrillin 1, a structural glycoprotein in the extracellular matrix. Fibrillin helps organize elastic fibers, so when it's faulty, tissues lose their architectural integrity. Fun fact that's actually not very fun if you have it. There are over 1,000 different mutations of the FBN1 gene. That's one of the reasons Marfan syndrome looks different from patient to patient. This is not a one mutation fits all situation. So bottom line, weak fibrillin means weak elastic fibers, and weak elastic fibers in high pressure systems like the aorta are a bad combination. This slide highlights the classical physical and clinical features of Marfan syndrome, many of which you can pick up just by looking at the patient. Patients are typically tall, with long arms, long legs, and long tapering fingers, a feature called arachnodactyly. Think built for basketball, but without the coordination or joint stability to enjoy it. Ligaments in the hands and feet are often lax, so patients may appear double jointed. This flexibility looks cool at parties, but is much less cool when joints start dislocating. Spinal deformities such as scoliosis and kyphosis are common, along with chest wall abnormalities like pectus excavatum and pectus carinatum. Basically, the sternum didn't get the memo about staying flat. One of the most distinctive findings is bilateral ectopia lentis, meaning dislocation of the ocular lens. This is almost exclusive to Marfan syndrome and is a huge diagnostic clue. Cardiovascular involvement is where things get serious. Patients can develop mitral valve prolapse and regurgitation, but the most dangerous complication is dilation of the ascending aorta. The leading cause of death in Marfan syndrome is aortic dissection or rupture, which is why early diagnosis and regular monitoring quite literally save lives. This slide reinforces one of my favorite pathology truths. When structure fails, the body leaves receipts. In connective tissue disorders like Marfan syndrome, the genetic defect shows up in very predictable anatomic patterns. You can often tell what's wrong before you even see a lab result. Chest wall deformities like pectus excavatum, where the sternum caves in, and pectus carinatum, where it sticks out, are classic examples of how altered connective tissue affects skeletal development. Long fingers, spinal curvature, joint laxicity, all of these trace back to defective elastic fibers and abnormal extracellular matrix organization. Some worthless but interesting knowledge. Abraham Lincoln is often speculated to have had Marfan syndrome based on his physical features. There's no definitive proof, but pathologists love a good historical differential diagnosis. The same structural weakness that affects the skeleton often also compromises the aortic wall, making it prone to dilation and dissection. So when we say structure tells the story, this is what we mean. Genetics drives structure, structure drives pathology, and pathology explains the clinical picture. And that brings us to the end of Lecture 3, Part 1, which means congratulations, your brain has officially done enough heavy lifting for one sitting. At this point, you've been introduced to chromosomal abnormalities, mutations, epigenetic changes, and inherited connective tissue disorders. And if some of those terms are starting to blur together, that's completely normal. That's not confusion, that's learning in progress. This is a good stopping point because everything we covered builds the foundation for what's coming next. Think of this lecture as setting up the scaffolding before we start hanging specific diseases and pathways on it. So take a break. Stand up, stretch, hydrate, blink a few times, and maybe remind yourself that you are not personally responsible for remembering all 1,000 FBN1 mutations. When we come back for Part 2, we'll keep layering onto this framework and start making even more clinical connections because pathology never misses an opportunity to escalate. Until then, step away from the screen for a bit. Your neurons deserve it.